In this video, we will discuss how the sialic acids are formed in the mammalian cells and how they are recycled back. That can be used again in this during this carbohydrate structures formation that contains sialic acid. So this sialic acid production begins in the cytosol, where this UDP glycnec is react to form mannec, and after that it is this ATP conversion, ATP to ADP conversion leads to the formation of mannec 6 phosphate, and this first two step is the rate limiting step. After that this mannec 6 phosphate is converted into the new 5 AC or sialic acid 9 phosphate through this NANS reaction, and thereafter this new 5 AC 9 phosphate is again converted into the new 5 AC, which is sialic acid, through this NANP reaction. And this process occurs in the cytosols, and once the sialic acids are formed in the cytosols, they will then move into the nucleus, where with the help of this CMAS reaction the CMP is attached to the sialic acid, and once this CMP sialic acids are formed, they can then be transferred into the Golgi, where with the help of different glycotransferase this sialic acid can be transferred to the carbohydrate structures, and based on the final structures, the glycoproteins proteoglycans or glycolipids can be directed to its final location at the Golgi at the trans Golgi network. And when they reach to its final location, such as these glycolipids, when glycoproteins and proteoglycans are reached at the cell surface, then then the sialic acids part are exposed to this extracellular matrix, and there with the sometimes this neuraminidase can cleaves off the sialic acids, whereas there are some enzymes that can cleave off complete glycolipids that can be seen here. And from this stage, the sialic acids that are freely available in the extracellular matrix, and also these carbohydrates contain sialic acids that are freely available in the extracellular matrix. They can interact with this Sialic receptors. Sialic receptors has an affinity towards the sialic acids, so they can detect the sialic acids and help in the endocytosis of these carbohydrate structures. And during this process, the carbohydrate structures that contain sialic acids or the sialic acids can be entered into the cytosols, and thereafter it will go into the lysosomes, and in the lysosome steps the carbohydrate structures can be recycled, and simultaneously along with the carbohydrate structures, this sialic acids will also be recycled back into the nucleus, where it can again react with the help of this CMAS to form CMP sialic acid, and this cycle will process. And this is how the sialic acid production occurs in the case of mammalian cells. And since there is a different kind of steps that are involved for the sialic acid productions, and the disruption in any of these steps could lead to the change in the sialic acid structures in the case of and that might lead to several problems. And in the case of pancreatic cancers, there is a lot of evidence that the sialic acids are overly expressed. Sialic acids are overly expressed on the cell surface of the tumor cells. So in that case, it is possible that there could be some kind of short circuiting occurs in the process that might lead to the overexpression of the sialic acids, and this rate limiting step the rate limiting step would have been over accelerated somehow in the case of, might be over accelerated somehow in the case of tumor cells, because there are a lot of metabolic reprogramming occurs in the tumor cells. So that could be one of the reasons why the sialic acids are overly expressed in the case of tumor cells. And also, since the tumor microenvironments are different, in the case of pancreatic tumors, it might be possible that the recycling step might be slowed down, because there could not be the rate of recycling could be slowed down. One of the reasons could be because of less cleavage of the sialic acids at the cell surface. So since the sialic acids on the overexpression of sialic acids helps the tumors from immune evasion, at the same time promotes proliferation and uncontrolled growth rate. So and this is possible because of the tumor microenvironment that is formed across the uh, pancreatic tumors or any other solid tumors, where the sialic acids are overly expressed on the cell surface. So this could be some of the possibilities, but not sure. We are not 100% sure that these are the exact changes. That still needs to be investigated. So I hope you got a basic idea how the sialic acids production occurs, and how this recycling of the sialic acids occur within the mammalian cells. If you don't understand about this sialic acid productions and its recycling in mammalian cells in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.